We are collaborated as a team of 9 members and done this video with the help of PowerPoint presentation. This video is to know about the past and present times of 65 roses. So why 65 roses? The 65 roses story dates back to 1965 when an observant, a small kid who was a 4-year-old, hearing the name of his disease for the first time, pronounced cystic fibrosis as 65 roses. Today, 65 roses is a term often used by young children with cystic fibrosis to pronounce the name of their disease. So let's get into the topic. Cystic fibrosis is an inherited autosomal recessive genetic disorder caused due to mutations in CFTR gene, which causes the cystic fibrosis transmembrane conductance regulator protein to become dysfunctional. Mutations of CF is usually located on chromosome number 7, which is in long arm. CFTR protein is responsible for transport of chloride ions in and out of cells, which regulates, balances chloride and water that produce watery mucus that helps cilia to discharge waste outside. Mutant CFTR is less able to transport chloride out of cells and draws water inside. This affects the function of cilia, which creates an environment prone to various bacterial infection in many organs, mainly in lungs. You can find the airways of normal people and people with CF. The various parts of our body affected CF is explained here.
Let us discuss about the early times case study. Cystic fibrosis was discovered by Dr. Dorothy Herring, a pathologist. She termed the disease as cystic fibrosis of pancreas, which was previously known as mucosividosis. The first modern description of the disease of clinical and pathological study for 49 children with pancreatic fibrosis was made in 1938 by her. She found the characteristics of cystic fibrosis in 1938 and 1942 and that it is caused by a recessive mutant gene. In 1950s, people with recessive form of cystic fibrosis rarely lived past age 5. In 50s and 60s, the treatments like pancreatic enzymes, airway clearance technique, anti-pseudomonal and anti-staphylococcal antibiotics increased the average life expectancy to age 15. In 70s and 90s, due to the treatments like lung transplants, DNAs, life expectancy jumped to 31 years. Inheritance pattern. Autosomal dominant. Each affected person usually has one affected parent. A copy of gene from any parent that transfer the disease. Example, Huntington's disease. Autosomal recessive. Both parents also affected are carriers. You need both copies of mutant gene to cause the disease. Example, sickle cell anemia. X-linked dominant. Frequently affects female. All daughters and no sons of an affected man will be affected. If mother is affected, all her offsprings are affected. Example, hypocathenic rickets. X-linked recessive. Frequently affects male. Example, hemophilia A. Then what about cystic fibrosis? Cystic fibrosis is an autosomal recessive disorder which affects both male and female equally. 1 in 4 persons with parents who are carriers of cystic fibrosis have greater chances to have the disease.
Genetic mutation. Genetic mutation occurs in a genome when a single base pair is added, substituted, or deleted that have various functional consequences like change in gene expression or alteration in encoded protein. It is classified into 2 types. Point mutation. It substitutes one nucleotide to other that produces altered protein. In frame-shift mutation, a single nucleotide is added, deleted, that also affects the reading of a base sequence and produces altered protein. For better understanding, we shall watch the below video.
When they start, when they stop, and determines how long the chain will be. So what happens when things go wrong? When errors occur in the DNA code, It is referred to as a mutation. The two main types of mutations are point mutations and frameshift mutations. A point mutation is where one of the three bases is replaced with a different base. Sometimes these mutations have minimal effect, but other times they can significantly alter the structure of the protein, depending on how different the new amino acid is from the original. The new base may also alter the protein by creating a stop signal too early in the amino acid chain. Mutations like this often render the protein completely ineffective. A frameshift mutation happens when one or more of the bases are inserted or deleted. Since bases are read in groups of 3, their addition or deletion in anything other than multiples of 3 disrupts the reading frame sequence. When this happens, the entire code following the addition or deletion will be read incorrectly. This could result in the wrong amino acid being coded, thereby changing the protein's shape and function.
Cystic fibrosis mutation. Mutation in CFTR gene leads to reduced to no function of CFTR protein. CF-causing mutations are divided to 5 classes based on the impact on protein production, that is, protein production mutations, protein processing mutations, gating mutations, conduction mutation, and insufficient protein mutation. In the first class, the CFTR gene is not read, assessed properly, and little to no CFTR protein is produced. In the second class, CFTR protein is created but misfolded, keeping it from reaching the cell surface. In third class, CFTR protein created and reaches the cell surface, but the gate does not work. In the fourth class, CFTR protein is produced and reaches the cell surface, but the opening of CFTR protein ion channel is faulty. In the fifth class, CFTR protein are produced in insufficient quantity. Frequency of occurrence. Cystic fibrosis is predominantly can be found in white European ancestry that affects 1 in 2,500 people, and it is less common in other ethnic groups. Now we can find images related to affected people.
Now let's recap this session with Q&A. Question 1. Where does cystic fibrosis affects the most? A. Respiratory and lungs. B. Digestive and respiratory system. C. Bones and skins. D. Pancreas. Question 2. Cystic fibrosis caused due to A. Recessive sex-linked gene on X chromosome. B. Dominant sex-linked gene on X chromosome. C. Dominant autosomal gene on chromosome 11. D. Recessive autosomal gene on chromosome 7. Question 3. How does cystic fibrosis is diagnosed? A. Sweat test. B. Blood test. C. Urine test. D. Both A and B.
Let us look into case study. Patient named Nicholas. Nicholas was affected and diagnosed with cystic fibrosis at the age of 1. The health issues is that he was born with blocked intestine and was immediately transferred to Cincinnati Children's Hospital for care by the top gastroenterology team, and he was fed with nutrition through a tube until his bowel were properly working.
Samantha was diagnosed with cystic fibrosis at age 3 and had been under several antibiotics and treatments. She is a firm believer in exercise, which kept her lungs healthy through the late 20s with the help of medical advancement. Originally from Wisconsin, she has lived in California since 2005. She has a bachelor's degree and a stable job. But at the age of 31, her lungs collapsed and undergone double lung transplant, which helped her live next 5 years peacefully without any treatment. Then came another fight in spring of 2016 that she developed chronic rejection and had to be hospitalized. But on October 11, 2017, she managed to receive another lung transplant and has been doing well ever since. Cure in process. The developments in the sphere of science increased the life expectancy of people with cystic fibrosis from 20s to 50s. Doctors and scientists are working on developing cure using methods like gene therapy and gene editing techniques, which are in early stages.
One possible way to restore CFTR protein is gene transfer. Gene transfer would work by inserting a normal CFTR DNA sequence into the cell so that it can make functional CFTR protein. Gene therapy could be inhaled or given through an IV. However, getting DNA into lungs is challenging. Cells are surrounded by protective membranes that are designed to keep out foreign objects. And in the lungs of people with CF, cells are also covered with a thick layer of mucus. Specialized delivery mechanisms called vectors are being developed which would take the new correct CFTR gene into the cells. There are multiple types of vectors derived from viruses or synthetic molecules. Gene transfer could potentially work on anyone with CF, no matter what kind of mutation they have. Gene editing in cystic fibrosis would repair the CFTR gene mutation in the person's own DNA. There are multiple gene editing techniques including CRISPR-Cas9, zinc finger nucleases, transcription activator-like effector nucleases, and others. Each of these techniques remove the mutations from the gene. To repair the damage, the cell puts in the correct DNA sequence for a functional CFTR gene. Gene editing can be done with cells inside the lung or in cells that have been taken out, corrected in the lab, and put back into the lung. Gene editing can either be tailored for each specific CF mutation or designed to correct most or all mutations. Gene editing changes the DNA permanently. So if CFTR mutations in stem cells can be edited, all daughter cells derived from the edited stem cells will make correct CFTR protein, making repeated treatments unnecessary.
This presentation was done by members of Team